We have
Good morning, everyone.
the
Welcome to STTP
pardon.
training program day 2.
Can you please unmute?
Please continue.
This is the second day of the lecture series or the FTP program. We have quite successfully ended the first day, and the first session of the second day. The title of the talk is Biomolecules, its structure for function. So this is given by me, and actually I think I do not need any introduction because I am a faculty of this university and I am working in the structural biology field. So, as the title of the particular FDP is Biology for Mankind, so we will start with a little bit. What is biology as per the dictionary? The definition of biology is basically it's a branch of knowledge that deals with the living organisms. So if we consider the architecture of biology, so the biology is basically says the cell is acting as the basic unit, genes actually acting as the basic unit of hereditary, and evolution basically acts as an engine that propels the evolution and the new species. So evolution we have yesterday understood little bit from the variations and others. So in a nutshell, we have to say the gene is acting as mainly the guardian that drives the process, regulatory function, and makes the information to act as a living cell alive. So in a cartoon representation, we find that the cell, which is the basic unit of life, then it goes to nucleus and genes, the chromosomes. We have yesterday told what is chromosome and DNA. And then we have to go for the next one. What are the basic parts on which the cell functions? Based on the function of the cell, if we divide its structure, just like a building, if we consider a building, the bricks are the unit. Similarly, here, in case of the cell, what we find is basically nothing but we are getting this DNA chromosome. What is the basic unit is the nucleotide, and we all know the nucleotides are basically the constituents or made of the three parts. Then we find the plasma membrane, where is a lipid bilayer and the proteins are there, and the amino acids are the building blocks. And the third part, which make the cell wall and other things, that was the carbohydrate sugar. So when we consider this particular things, that is the nucleotides, amino acids, and sugar or the lipid, the fundamental question is what is the basic constituents of these all biomolecules? Long ago, we find that there are Newland, there are other persons are there who are trying to organize the elements that were found in nature. Most probably, and the Dmitry Mendeleev, the Russian scientist, who has basically organized the elements based on their property. And as per that time, only 53 to 60 elements were there. He started with working on 53 and then ended with 60. And he made these groupings, group 1, group 2, group 3, like that. Group 1 is called the alkali metals. Then there's called the alkaline earth metals, then the non-metals, gases, etc. And during his time, there is no information regarding the noble gases like helium, neon, xenon, argon, etc. But the work he started, and that ended with the new modern periodic table, where we find there are 118 elements at there. When there are 118 elements and there, and you find there are basically they are colored based on their chemical properties. Now the question is, do all these elements are important for the formation of the biomolecules? Now, if we go to the definition of the biomolecules from the earlier days, we have to say, we have to remember, recapitulate the name of Volher, who first say that biomolecules are not only the molecules which are obtained from the biological sources. Actually, by the definition of biomolecules, we can say the biological molecules or biomolecules are those which can be synthesized by cell, which can be obtained from the different organisms of the system. So they are. But actually, Volher, by the conversion of urea to ammonium cyanide, shows that the biomolecules and the inorganic molecules, they can be shifted. But however, till date, we are considering as biomolecules for those molecules which are basically obtained from the biological sources. And if we consider the basic constituent of the elements of these biomolecules, we find that these are mostly organic compounds, and based on the classification of the organic compounds and its structure, the basic constituents are carbon and hydrogen. And with this carbon and hydrogen, there are oxygen, there are nitrogen, there are phosphorus, and sometimes there are sulfurs. So the biomolecules, if we consider, we find that the main constituents of the biomolecules are nothing but carbon, hydrogen, nitrogen, oxygen, phosphorus, and sometimes sulfur. Though in some other cases, to can argue selenium, etc., is found, but these are the main basic constituents. So what we find is how to represent this, biomolecules. There is a color code. When we are talking about a biomolecules to represent, this is called the CPK. Dear Prem, can you see the word color these lines are showing?
Yes, sir.
The color code of this CPK, it is coming.
The color code is not displaying, sir.
So how we can remove this part? Actually, I want to.
Sir, you can do one thing. You just close the share option. You close the sharing option, sir.
Stop sharing.
Yes, sir. Stop sharing.
Okay.
So now you just click on the share screen and you just check on the first option. After
This
clicking
screen.
the share, you just click on the first option, sir. Screen. Click on the share screen button, then click on the first option, and then share it, sir.
This is only showing the share, nothing more, nothing else.
Sir, share screen clicking after that first
Only
option.
only share screen. Okay. Then
Sir, then first option screen.
share sound objective is there.
The whiteboard is displayed at the top. Whiteboard,
Yeah,
I think.
whiteboard is displayed.
Yes, sir, at the left, see, there is screen only. Written screen.
A screen is there.
Yes, I just click on the screen and after that share it.
And now I have to share. If I share,
Just share, but share button se click.
then again it is coming the same.
Same thing. So you go to the desktop, sir. Minimize it, sir.
Desktop.
Yes, sir. So it is not showing your desktop. So your desktop should be visible, sir.
Yeah.
For that,
Can you now see?
no, sir. It is not nothing displayed, sir. So you just share the screen once again by using the screen option, sir. Click on the share screen, then click on the screen option beside the whiteboard. And after that, click on the share.
This one, then share screen, then screen option,
Screen
then
option, yes, sir. Then share button.
then share button.
Directly share button, yes, sir.
Okay.
Yes, sir. Yes, sir. It is now visible.
Still it is coming.
So can you show me the desktop, sir?
Huh?
Can you show the desktop, sir?
Desktop. Show the desktop.
Yes, it is showing, sir. You can do, sir. Editing. You can do, sir.
What I can do? I can start with this one.
Yes, I start. Yes, I start the presentation. Yes, sir.
Again,
So
it is
color
coming.
code is not displaying.
Yeah. Actually, the mute, stop sharing, that is coming.
Sir,
So what to do?
so is there any double screen that you want to show, or there is some written textual information?
If I send you this thing, can it be possible?
Yes, sir. Yes, sir. Just send me, sir.
If I
Let me
send
check
you
from my
the,
end. Yes, sir.
huh?
Yes, sir.
If I send you this, I'm forwarding this because if the write-up is not coming, then it will be a problem. This is, it was sent. It was sent. Have you got it?
Sir, I'm checking.
Or if I go in this way, can you see it? The color convention. Is it visible? Participants? Hello?
Yes, sir.
Yes, it is visible like red, blue, and yellow.
Yeah. But now, color convention CPK model
No, there we cannot.
cannot. Okay.
Red is hydrogen, but this we can in the model it is the red, white, and blue.
Yeah, I pardon. I have seen this thing, but actually that part is a little bit creating problem because otherwise I am giving not justice to the persons who have done this. It is still coming this way.
Sir, like are you talking like the top part, the starting the example?
Yeah. Yeah.
Right, it is red, obvious, right? Then it is followed by the white, that's a ring.
No. Can
And
you
down,
read the color? Can you read the color code? Can you read the CPK model? Can you read it?
Yes, it is readable,
It's
sir.
visible, sir.
It is readable.
It is readable.
example,
Color code.
Yes, sir. It is visible.
Sir, are you talking about the written description beside the color code?
Inside the color code top, there is a title color coding CPK model. Can you read the word CPK?
yes.
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Sir, hydrogen for white, oxygen for red.
No. Over hydrogen, there is a color, there is a title.
Yes,
Heading
it is
is reading.
readable, sir. Color convention, CPK
Yeah.
model.
Yeah. Oh, then it's okay. Actually, in also.
Sir,
So this is
it's visible, clearly
okay.
visible color conventions and CPK model.
Okay. Thank you. Actually, in my screen, I cannot see it. It is showing the stop sharing. Your screen is shared, and then so many things are there. Okay. So this is a color convention. When you are justifying, you are showing the biomolecules, there is a color convention, and this color convention is called the CPK model. This C, P, and K, these are the three names of the scientists who are all Nobel laureate. The C is E.J. Corey, P is Linus Pauling, who received the Nobel Prize for 2 times, and K is Coulter. In 1950 onwards, Corey and Pauling initially gives this hydrogen is white, oxygen is red, chlorine is green, and nitrogen is blue, carbon is gray, sulfur is yellow, phosphorus orange, and others. So what we have to keep it in mind is basically when we say this thing, this is the what I am showing here. In this particular case, an example of the atom color convention used with our amino acids, because this is we will work with the biomolecules. So oxygen, this is an example of the tyrosine molecule. This tyrosine molecule you see, this is the NH2 main chain. This is the gray one, carbon, the C alpha carbon. And this one is the carbonyl carbon and the carbonyl oxygen. And this part, so the ring part is nothing but the main chain part. And this other part is the phenyl ring with the OH group is the tyrosine. So this model is the given by Corey and Pauling. And then there is another one which is given by Carlton, that is called the space filling model. And in this particular space filling model, we find that there is carbon gray, nitrogen blue, oxygen red, and there are yellow. These are the sulfur-sulfur disulfide linkage. So this space filling model is given by Carlton, and altogether nowadays this is called the CPK model. So now when these constituents are present, the immediate question comes into our mind, how these are interacting or how these two are joined together. So there are the interactions are basically 3 types of interactions we have, or mainly we can classify 2 types. One is called the covalent interaction and the electrostatic interaction. In case of electrostatic interaction, the bond is called the ionic bond. The example is sodium chloride. We know these things. The covalent interaction, methane is there. And there are weak electrostatic interactions. And along with that, there is a dative bond or the coordinate bond is there in case of the ammonium chloride. This is a simple class 10 question. Name one molecule where you can find all kinds of bonding. This is ammonium chloride. You find that ammonium NH4 plus and Cl minus, they are connected with ionic bond. Now the NH4 formation, that is NH3, is dative bonding coordinate to H plus. So this is the NH4 plus is formed by a dative bonding. And if we consider the NH3, there is a covalent bonding is there. So when we are considering whatever the small molecule or whatever the macromolecule, it doesn't bother or it doesn't matter. The interaction is always these three types. Now we are coming to the types of different biomolecules. If you can remember my third slide where we find that the cell and divided into this one, if I go back to our this third slide, so you can find that thing is basically the cell. They are constituent of the three, mainly four parts are the nucleotides, amino acid, lipids, and the sugar. So we will find these things. Now, if we go for these particular things, these are the four category, the biomolecules mainly can be divided. So in case of this, we find that lipid, these are the two part is this. So what we are getting in case of lipid, we find this part is nothing but this is basically this part. If we see, this is the hydrophobic part where we will get only the carbon and hydrogen aliphatic long chain. And this is the part where we find this is basically the CO2H group. This is the polar group. Then we come to the carbohydrate where we find there's a number of molecules, number of functional groups are there, and mostly all the carbon atoms. So these are basically, this is the open chain, and this open chain where we find, so this is the open chain where is the constituent of the carbon, and all are the OH groups are there. So these are the functional groups, polar functional groups, and along with that there is a CHO group. So this is called the open chain structure of carbohydrate, and its name is aldohexose. And here in the first, so these all are chiral carbon, and this the OH is in the right hand side, so it is called the D. There are two types, the D and L. So this is a D-aldohexose. And what we find in case there is another way of representation where this OH interacts with this CHO group and they are forming a cycle, and this is called the Howard presentation. And where the OH may be up or OH may be down. So this nomenclature is basically we are writing the D-aldohexose. So this is D-aldohexose, and here we are writing another one before D, that is beta, because this OH group is up. So this is beta D-aldohexose. And then we come to this next one. This is the nucleic acid, and the nucleic acids are the constituent of the base, that is the bases, the ATGC base. They're the sugar moieties, and the sugar are of two types, ribose and deoxyribose, and then they are connected with the phosphate, that is the phosphodiester linkages. So this is called the nucleotide. And the last but not least is the proteins. So when we are going to this, the carbohydrate, this carbohydrates, actually this is nothing but a recapitulation. This is the monosaccharides, where only one unit is there. Then there is a oligosaccharide where 2 to 10, number of units are connected, and this particular linkage is called the glycosidic link. So in case of polysaccharide, more than 10 units are connected, and these are connected, all are connected by. So if we go in these particular cases, here we find that the main constituents are carbon and hydrogen and oxygen. Here also we find there is a carbon, hydrogen, and oxygen. In case of the nucleoside and nucleic acid and the protein, that is DNA and RNA, we find that along with the carbon, hydrogen, and oxygen, we are getting nitrogen and we are getting others. So in case of carbohydrate, the name is actually coined from hydrated carbon, because if you see the structure, we can write it as CxH2O whole Y in a general form. CxH2O whole Y. But although it is not that thing, initially people after the element detection, they are saying that thing, so they say that hydrated carbon, so this called the carbohydrate. So one of the very important example of these carbohydrate polysaccharide is pectin, which has a huge use and which is the main constituent of the cell wall formation. So next come to the lipid. Lipid can be divided into the simple complex, and lipid has a hydrophobic tail and hydrophilic head. And there is a basically in the hydrophilic part, the phosphate group there, so they are called the phospholipids. And this is the main constituents of our cell membrane. And this is the lipid bilayer, where in case of the lipid bilayer, we find that this part is the hydrophilic part, this part is the hydrophilic part, and this is the hydrophobic part. They are basically forming, and this is basically the complete structure of the cell membrane where we find. And in this particular case of the cell membrane, if we consider this one, we find that this is a protein which exocyclic part, what is inner part is there, and this part of the protein which is very important, that is called the transmembrane part. So similarly, in case of the basic nucleic acid, we find the sugar. There are two types. I have already told you this is the 2' position. In this case, this is the 2 position. And so what we are getting is that if we have a OH group is there, so this is called ribose, and this one is that this is not this. So what we are getting, we find that this is basically the deoxyribose. In case of the DNA and in case of RNA, the basic difference is not only the single and the double strand, mainly in the difference in the ribose sugar. So we have, this is the complete structure which we categorically see this. Now come to the, after DNA, we have the RNA, and this RNA, this is basically mRNA, messenger RNA. There is a codons part is there. This part is the codon part, which we have to work with. And this is the tRNA. And this tRNA is basically considering the anticodon, and there are mRNAs. Now come to the main part, which I am interested and which I will work with, the amino acid. So there are 20 different amino acids are found, and in case of the 20 different amino acids, we found there is a NH2. This is the main chain of the main part of the amino acid, and this all the 20 amino acids differ with the organ. And this is as because this is a chiral carbon, there are two types, D and L nomenclature is there, and in case of D and L, nature has cleverly taken only the L part. And when the two amino acids combine, there is an elimination of water, we are generating a peptide bond. And this peptide bond, basically, this is actually our journey will start from here. We find that there are 2 points. If we have 2 points, we can draw a straight line. If we have 3 points, we can draw an angle. But if there is a plane, if there is a plane and there is another plane, and the angle between two planes is called the dihedral angle. So when we are going to talk about this, a protein structure, protein conformation, etc., we have to keep it in our mind. We are talking about a dihedral angle. And this dihedral angle is also called in another name, it is called the torsion angle. So what is this dihedral angle? This dihedral angle is basically, there are 3 types of dihedral angle we find. This is the dihedral angle between this one. If we find between the CO and N, this is called the omega. Then N and C alpha, this is the phi, and C alpha CO, this is chi. Again, if we proceed, it will be omega. Again, it is phi, and then you will find it is chi. So a peptide, which is a linear combination of the amino acid, they can represent the whole peptide main chain by the three dihedral angle, that is omega and phi and psi. So how we will get this thing, omega, phi, psi? So omega and phi, these are the two common points which we are talking about this one. And there is one extra in the back. So if we talk about omega, which is a dihedral angle between the C and N, so we have to take, this is one plane we have to consider, C alpha, this is the ith residue if we consider, and this one is another one. This one is another one. So this dihedral angle, so this is I plus 1. So this is the I plus 1. So it is the dihedral angle or the angle between two planes of the I and I plus 1, where the C carbonyl and the nitrogen is the common 2 points. If we go to phi, then N and C alpha will be the two common points, and the one left and one right will be the another. So if we consider the protein structure like the nucleotide, and where we find the DNA is a structure basically constituents of the 4 nucleic acid base along with the ribose or deoxyribose and the phosphates. Similarly, the protein structure, only they are constituents of the amino acid. There are no other parts of it. If the amino acid, the linearly combined, just like if we consider you are making a garland with some flower. If you are not making a shape, if you were just doing organizing this one after another, similarly, you are threading this thing and you are ending with a primary structure. So the primary structure is basically, when we are talking about, it is basically considering only the omega. So when this one is now locally, so if you consider in this way, if you are just making one brick, another one, another one, and you are making a line, but the line cannot be a longer and longer because you have a restriction of your space. So what you have to do, you have to fold it, and if you fold it, so you are getting a plane, and that plane is basically the dihedral angle. And similarly, in case of the linear structure of the protein, it cannot go along it longer and longer. So due to the space crunch, it has to fold. It has to turn, it has to take a shape. And once it has to take a shape, it will basically give a, it has to take the phi and psi. And what is we are getting? So this phi and psi will give you, depending on that, mainly the secondary structure. And this secondary structure, when we interact through space, so the secondary structures are locally formed. I am coming to that. And when the secondary structures is interacting and they are arranging in some way, so that is giving a tertiary structure, and the tertiary structure, again, it will be forming a domain-like thing. It's a basically quaternary. So we can simply take an example of our apartment or flats. So what we are thinking about, all the apartment which in a complex or in a flat multi-storied building, all are almost similar, and the main constituents are brick. But in case of the longer chain, the linear chain of the room, this is the primary structure. But once it get a shape of the square or the triangle, etc., then you are getting basically a secondary structure. And in case of the different secondary structures, that is your bedroom, that is your kitchen, that is your living room, that is your toilet, all are having either square shape or the pentagon shape, or these are the different shapes. But the main constituents are bricks, but they accumulated together to give rise to a function. And one is bedroom is related to your living room is related to your kitchen is related to your toilet. So they make an arrangement, and this arrangement is called basically the tertiary structure. And once the tertiary structure is now depending on the function that is called the domains and the quaternary structure, and the whole multiplex or the whole apartment or the whole part of your multi-storied building will act as basically the quaternary structure. Now, what we are doing this thing in case of the biology, this biological system, the very important and the common part is central dogma, where the flow of information takes place. Actually, even yesterday you were talking, you were asking whether the genetic information, yes, genetic information is important because that is within the DNA. And that is transcription. Through transcription forms the mRNA, and the mRNA, the information is going to translate to protein. So when we are talking about a central dogma, the central dogma is nothing but it is a DNA to protein through RNA, and where the genetic information is translated, and now they are doing the function. So what I am trying to show, I am trying to show how these amino acids are going to form. Just a minute. Just a minute. Prem, how can I get back to this arrow one? Okay, but this is not coming.
Sir, pointer option. Second one, sir, pointer option.
Pointer
Yes.
option. I want the
Sir, laser pointer. Yes, sir.
laser pointer. So it
Yes, sir.
goes the laser, but actually there is a movie is here. So if I click on the movie, it will go like this.
Sir, actually here is some video.
Yes. A video is there.
So video, sir, for that you play it, sir. That video.
Where I have to play?
Sir, just click
So
on the slide. It
yeah, I
will
click on
automatically
the
play.
side, but it goes like this. Actually, I have to change the pointer.
Yes.
Okay. Mm. Well, I have to come back to arrow. Okay. Yes, I'm showing. I think I can.
So you have to come back to arrow, no, sir?
Yeah. I have to come back to arrow.
Sir, then just screen share, sir, once again.
Okay. Can you see this?
Sir, nothing is displayed now.
Nothing is displayed now.
Translation is initiated by formation of an initiation complex.
Okay, can you see now?
Yes, sir. Now it is visible, sir.
Eukaryotic cells. Translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are involved but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids combine. One is called the peptidal or P site, and the other is called the acceptor or A site. There is also a third
Sir,
site called the
Is it
exit
visible?
or E site, where
sir, it is
transfer
visible,
RNAs
but
are
can
released.
you play it from
The
the
initiating
beginning?
Yes,
transfer
sir.
RNA
Pardon?
It
carrying
was not
formal
audible, sir.
methionine binds
Somebody
to
is telling that to play from starting.
Yeah.
Yes, sir. Because
Yeah,
from
yeah.
the beginning, if you can play. Yeah.
Yeah, sure. 1 second, I'm.
Thank you, sir.
Can you see
In
now?
prokaryotic cells, translation
Yes,
is
sir.
initiated by formation of an initiation
Yes, sir.
complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids combine. One is called the peptidal or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay. Have you heard it? Hello?
Yes,
Yes,
sir.
sir.
Yes, sir.
So this is basically what we have come to know, how the protein biosynthesis is going to occur. Okay, so this one.
In prokaryotic cells,
Once again, I'm showing
is
it.
initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptide, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
So this is the basic part, how the protein biosynthesis is occurred during in our cell. And actually, I hope everyone can tell why the codon number is 64. Why the codons are triplet and this is the 64? Because the 4 nucleotide bases are there and 20 amino acids are there. So it is just a mapping. 4 can take maximum 4. So 16 amino acids are left. So it is cannot be defined. And then another 4, that is the 4 to the power 2. This is a 16. If this is, that means ATGC, then come AA, AT, AG, AC, etc. It's a 4 by 4 matrix. So it will generate the 16, and the 16 can take 1 amino acid, 16 amino acids, still 4 are left. So then come to the 4 to the power 3. That is a 4 is to 4 into 4, 4 cube 64. And that's why the codon degeneracy comes. So 1 amino acids, because the next question is why we look for proteins. Because basically that was the thing why nature is taking the codon degeneracy, not the amino acid. If we consider the protein, if we consider the function of cell, the proteins are the mainly that are the machines for making the cell function. This is a very important role. And from the RNA or DNA level, it is not possible to have an accurate level or the accurate prediction for the protein levels. And the third one, why we have to know the structure and the function of the protein? Because we have to go for the target of the drugs. So I am seeing the whole thing just like a football match. The 11 football players, they are playing on the ground. They can make you win or they can make you defeat. So this 11 football player, they are basically the proteins. And the coach who are giving the all kind of information, that is the DNA and RNA. So if you find there is a different function, if you take the goalkeeper from the goalkeeper position to the striker position, and if you take the forward to the goalkeeper position, there is some distribution difference, and this you will be mismatching, and that will not do the proper function. So why we are interested in protein? Because it does the function. What kind of functions? Everyone knows that they act as a biological catalyst called enzyme. They are important for transport of oxygen. They are accumulating the different informations. They are occurring as a cellular defense. They are making the structural basic part like silk, collagen, etc. So they are also acting as a hormones like insulin and etc. regulatory hormones. So if we consider, although the DNA is taking the information, but the main key player for making the biological system active is protein. So what we have to know, already I have shown you the protein is a structural hierarchy, the linear sequence or the primary structure or the secondary structure, which is local formation. Now I am coming to that. The tertiary structure, that is the three-dimensional conformation, and the quaternary structure, which is basically combination of the multiple tertiary and the whole circle. So how we have to do this? Now, when being a structural biologist, someone yesterday told who was teaching in or who is teaching in techno, she said that she was working with the protein structure and etc. So it will be interesting for her, I think, that we are not talking about the amino acids. When we are talking about the protein structure, in case of the idea of the structural biologist, we are talking about the dihedral angle. And I have already defined the dihedral angle is basically, it's an angle between the two planes, and the three dihedral angle, omega, phi, and psi, which are the repetitive way in the main chain described. So the conformational flexibility. So when we are talking about the primary structures, we are not talking about any conformation. When we are talking, so what is a conformation? Now, there are two terms. One is called the conformation, and the second one is called the configuration. So what is configuration? We have talked about the D and L nomenclature. Capital D, capital N, that is the arrangement in three-dimensional space. But if we can see this thing, this is one of my part, and this is the single bond. If we rotate this thing around the single bond, we can find that thing. This is called conformation. So once we are talking about the conformation, this is a momentary arrangement arising due to the single bond rotation. And based on this single bond rotation, we are having these different torsion angles, and based on that, we find different secondary structures. So these secondary structures, they are two types, mainly regular secondary structures and irregular secondary. The regular secondary structures are alpha helices, beta strands, and the irregular secondary structures are tight turns, they are random coils, balls, etc. So this is in the left-hand part. I have shown you the secondary structure. This is the beta pleated sheet. This beta pleated sheet, the constituent of the main secondary structures are the beta strands, and the arrangement is depending on the arrangement, we will call it a sheet, antiparallel or parallel. And there is an alpha helix, the helicals, where I am coming to that what is helix, and there is another one is the random coil. So what we are showing this thing in a shortcut way, a structural biologist has basically shown the helix like this one. A strand is like an arrow. This is the N terminal and this is the C terminal. Why this is N terminal and why this is C terminal? Because we find that the electron density, the carbonyl carbon is more electron dense. So that arrowhead goes like this, and this is the loop region also. So when we are talking about, this is the famous G.N. Ramachandran Plot, the Indian scientist who has nominated several times for Nobel Prize, but cannot or couldn't get the Nobel Prize. But based only this particular phi and psi, there's two-dimensional map, which is giving a three-dimensional representation of the protein structure. We find that whole structural biology starts from here. Now, in case of this, if we start from here, this is a 0 degree. If we go like this is a 360 degree. But if you find this GN Ramachandran map, you find that there's a 0 and there is a plus 180 in the right-hand side and there is a minus 180 in the right-hand side. Why he has done this thing? Because if you can see this particular thing, in this particular thing, this is a circle, which is a 360 degree. We can easily divide it into two parts. The first right-hand part is a positive. So if you move to 0 to this way in the clockwise direction, you will get a plus 180. You can move to that way also, and you are reaching to 360. But if you go in a backward direction or the anti-clockwise, you will get basically 0 to minus 180. And now if you see this thing, this is a 60 degree and this is basically 300 degree. But if someone is asked the 60 and what is the relation of 300, it is very difficult or cumbersome. But if you see this is a plus 60 and this is basically a minus 60, then automatically you can see these are nothing but the mirror images to each other. So what we are getting in the two-dimensional plot, when Ramachandran initially he plotted the first paper in JMB, he plotted in 0 to 360 degree. Actually, that is as per the Linus-Pauling plot. But then in the next paper of the 68 with Shashi Shekharan and others, actually I'm fortunate enough to show this person, Sri Ramakrishnan and Shashi Shekharan, who are the key persons of GN Ramachandran plot, but I'm fortunate enough to see in IISc. So they said that they divided into this part and to get it basically to give an mirror image. And if you see this thing, there are the 4 quadrants are there. This is 0, 0. This first quadrant is where both the phi and psi is positive. In the second quadrant, phi is negative, but psi is positive. Then the third quadrant, psi and phi both are negative. And here the phi is positive, psi is negative. So the second quadrant and fourth quadrant are mirror image. First quadrant and third quadrant are mirror. And depending on the van der Waals interaction, that is the contact distance, they found that only these very short positions or short spaces are allowed. And these are called the allowed Ramachandran region, where the conformation of the phi and psi is. And actually, if you are asked, sometimes you will find even in book of Lellinger or etc., they are saying the definition of helix is nothing but it is an hydrogen bond. But the answer is no. Hydrogen bond is not the cause of formation of the secondary structure. Hydrogen bond is an effect. The cause of the secondary structure formation is basically the phi and psi. As the phi and psi, I'm coming to this one. I'm coming to this as the phi and psi takes in that way. Pardon? Hello? Hello?
Yes, sir.
Is there any problem?
No,
Should
sir.
I-
We have some background noise from
Okay.
Ruhi Mustaq, ma'am.
Okay.
So please continue,
So,
sir.
okay. So if we find that thing, the linear structures are there. But due to the taking of the phi and psi, you find that thing, this phi and psi. I'm coming to here. This is the around phi is minus 60 and psi is minus 50. We find that there is an I and I plus 4. The carbonyl and the nitrogen is very close to each other, so they can form the hydrogen bond. So the phi and psi is the direct cause, not the hydrogen bond. Hydrogen bond is the effect. Cause is the phi and psi. So what we are getting in this particular case, we find that there are beta pleated sheet, we find that the helices, and we find that there is a 20 amino acids are there. Now the question is, all these 20 amino acids are identical preference for this? Then the person name called Chow and Fashman in 1970s, basically in 1974, they published a paper and they show that no, the 20 amino acids have not equal preference for the secondary structure. They found that alanine, lysine, leucine, these are having the preference for the helices. Or the secondary structure, beta strand, is mainly preferred by the beta branched amino acid like isoleucine, like valine, etc., phenylalanine, etc. So they found that based on that, they have classified this thing and they said that B means strong and the strong breaker. And then the F means the strong former. So they found that Ella, the helix is F, capital F is strong former, and the propensity is 1.42. And if you find that methionine, it is 1.45. You find that glutamic acid 1.51. So that means these are strongly helix former. But if you find proline 0.57, that is capital B, the strong breaker. If you find that thing, glycine, that is a strong breaker. So what we find that in this particular bar plot, we find that depending on the 20 amino acid, the propensity for formation of helices and propensity for formation of the beta strands is different. So this is a clue for a peptide tailor who wants to design a peptide in case of the therapeutics for a helix or a strand, what amino acid has to be incorporated. Now, this is an example, direct example, which shows that if there is a proline is there, how it affects. If the proline is present, which is considered as a helix breaker, and if you consider this line is a touch line for 100% helix formation, you find that the jth residue has a lowering in helix formation compared to there where methionine a helix former is there. This particular case, the proline, if it is replaced, if it's present in the system, that is lowering the formation of the helix. So that is an example where we find basically proline is an helix breaker. The similar to that is the basically what we find in case of glycine. Now, what we find in case of the helix, the geometry, the structure when we are talking about, it is basically a screw. It's like a screw in physics, there is a pitch. Similarly, helix is a pitch, and it is basically pitch is equal to D into N, where N is equal to rise per turn, unit per rise in unit per turn, and D is the rise per repeating unit. So depending on this, the pitch, we find that there are different types of helices, and this is an example. That is the ring, that is the alpha helices, what we have written, alpha helices, 2.27 helices, that is a 310 helices, 5 helices. So we are having this. And out of this, we find that there are several helices, but the alpha helices is the most abundant and common. So this is the nature. You find that this is the left-handed helix and this is the right-handed helix. You can see these are the mirror image of each other. And this is the alpha helix. You find that this is a 310 helix, right-handed, this is an alpha helix, so you find the pitch is different. So I want to show you how, although they are phi and psi is very close, but depending on the orientation, depending on the arrangement, you can find a room 11 feet by 13 feet, that is also a rectangular, and 11 and 16 feet, that is also another rectangular. Both are rectangular, both are helices, but their pitch, their shape is little different. So I have already shown that alpha helix, which is the most common, this is a two-dimensional view, and then here you find that the R groups are exposed out, and that is called the Gauss interaction. So this is the way, or this is the reason for which we are finding that the alpha helices are most common. Here we find that 3.6 amino acid per turn, that is Dn is 3.6, pitch is 5.4 angstrom, and there's a hydrogen bond between the carbonyl I and nitrogen I plus 4. So there is a helix dipole is there, and typically the phi and psi is minus 60, minus 50. Now, in this particular case, we have already shown you that helix formation is a local. I am coming to that, whereas in case of the beta sheet, it is a non-local formation. Why? In case of the beta strand, if there is a movement in the similar direction, this is called the parallel beta sheet. But the hydrogen bond is intermolecular. Whereas in case of this one, even a single alpha helices is there is an intramolecular hydrogen bond is there. So when there is an intramolecular hydrogen bond is there, what we find is basically in case of this, we find that there is a I to I plus 4 hydrogen bond is there. And in this other case of the right-handed and the left-handed, whatever the thing, the parallel and the anti-parallel beta sheets or the mixed beta sheet, you require the two different strands. So if you give a dilution, how do you understand? If you give a dilution, these two are separated and you are losing the hydrogen bond. But in case of the alpha helices, whatever the concentration you are using, that doesn't bother, that doesn't matter, and actually the hydrogen bond exists. This is a typical example of the protein, and based on that, you find that this goes like this. So this is one structure, and this goes like this. This is called the antiparallel. This is also goes like this one. So this is a parallel. So this is basically a formation of the both beta sheet structures is there, immunoglobulin, and this is irabutyroxine. This is an example where we are having this. There are other things are there, which is beta turn. And if you consider the turn is a part of a helix which is not completed. So what we are having in case of the mostly important turn is the beta turn, where the number of residues will be important. A beta turn is basically, it is a high residues of I plus 1, I plus 2, and I plus 3. They do not form a particular helix, but a proper helix. These are like an horseshoe. These are like an oxbow type, where it is not completed. And if you see in this particular thing, I hope you can see this is a turn-like structure where you can have this. If you go for this, this is a helix, but you can have in this way, you can find this thing, this is a turn structure. And in case of turn structure, number of residues are present, and depending on that, there are different turns are there. Gamma turn, where the turn number of residues within the position is 2. The beta turn, which is the most important turn, where the number of residues are 4, and so on. So this is the total form of the Ramachandran plot, where we find that this is the parallel beta sheets. It is in the second quadrant, the beta sheet and beta strands are there. The collagen triple helices are there, whereas the right-handed twist beta sheets are there, anti-parallel beta sheets are there, and the 1 and 3, they are basically the mirror image to each other. If you take and just plane like this between plus 180 and plus 180 of I and psi, the third and the first quadrant, they are common to each other, mirror image to each other. So you are having in the third quadrant the right-handed alpha helix, and here you are getting the left-handed alpha helix. So up to this, is there any question? Up to this, is there any question?
Hello, sir.
Yeah.
Sir, can you tell me again that thing that why alpha helix is the common helix structure?
Okay. Why alpha helix is common structure? Actually, if I take a thing, this is basically, I'm showing it to here. Okay. This is called helical wheel model. So what we have shown you in this thing, this one. Okay. So in this particular case, I am drawing it here, so it will be easier for you to understand.
Yes.
Okay. So the alpha helix goes like this. This is a 3.6. This goes like this. Okay. So if you see this thing, these are the R groups. Okay. These are R groups. And now you see this is a 310 helix. So 310 helix means 3 residue per turn. So it goes 1, 2, 3, and this is the 4. This is 5, this is 6. So you find that 1 and 4 is there, 2 and 5 is there, and this is 3 and 7 is there. So this is called the eclipsing interaction. And this is the Gauss interaction is there. If you can remember the potential energy diagram of this thing, you will find that this is the eclipse, this is the Gauss, this is the anti, this is the, again, partial eclipse, this one is this, and this. So these are basically the Gauss interaction. Okay. And this is the partial eclipse interaction. And this is the eclipsing interaction between the side chain. So in case of 310, if you see this thing, this is 310 and this is alpha. So where in case of alpha, the Gauss means this one. This one is Gauss, where the angle is between 60 degrees. Okay. But in case of eclipsing, this is an angle, this is the 0 degree. Okay. So that means one, and can you see in the, do you have me in the screen? So
Yes,
these are the two. Okay. You can see this thing. So this is, and this angle is 60 degrees. So in case of alpha helices, what happens? These two dihedral angles, they are making a Gauss interaction. So they are not basically hindering. They are not sterically hindering each other. But in case of the 310 helix, it goes like this. This is an eclipsing interaction. So one is over another. That means energy becomes high. And I am not going into the protein folding. If the protein folding starts, then what we see is initially the 310 helix is formed, because you have to fix the 2 phi and psi. Whereas in case of, if I get the time, I will tell you this thing. If we, whereas in case of the 310 helix, you have to give the 3 phi and psi. So you can see like this, and I have this thing. This is, you can see this thing. In case of this, suppose this is an alpha helix, 1, 2, 3, and 4, and 5. I to I plus 4 hydrogen bond is there. Okay.
Yes.
So if you have this thing, so how many phi psi you have to fix? You have to fix the 3 pairs of phi and psi. Okay. But if you have a 310 helix, you find that 1, 2, 3, and 4. So you have to I plus 3 hydrogen bond is there. So that means the nucleation, in case of starting, you have to fix the 2 phi. Okay. So this is a concept called the kinetically controlled versus a thermodynamically controlled. So in case of a kinetically controlled, what you are getting, kinetically controlled means where you can easily understand, easily reach. That means the energy barrier. So you have to see a cinema, okay, movie, where the ticket price is 20, and another hall, INOX, where the ticket price is 100. So if you have 30 rupees, it is easier to go to any movie hall where you get the ticket with 30 rupees. But that may be not giving not so comfort, air conditions, and etc. But you can see the movie. But if you can spare the 100 rupees, you can go. So, but accumulating 100 rupees is not easier than accumulating 20 or 30 rupees. So when the protein folding starts from random coil, this is the 11th paradox. This is showing that initially you have to start with a low one where the energy is low. But once the energy is low, if you go to this one, this Ramachandran plot, there is alpha and tritane where there is no energy barrier. Okay. So after you are getting 20, 20, 20, 20, somehow once you reach the 100, you are changing. Initially you were here, but you change to this. And I have a paper in 2009 proteins where this is the first time we explain how this Ramachandran plot, from the concept of Ramachandran plot, written helix, initially it was saying that alpha helix and the random coil, it is a two-state transition. But no, it is not a two-state transition. It is an conformational ensembles are there, and where the written helix is a very, very important intermediate. And this is found experimentally. We have shown by NMR and all other things. If you can go through my paper in 2009 proteins, you will see that initially written helix is formed, and this written helix then grow and grow. Then suddenly you find this is to have accumulated 80 and 20. Suddenly you jump to from the suppose a Mitra all Roxy to the Inox. That is that. Okay. So the whole part is the energy. Whole part is the energy. How you? So this is basically, the term is called the nucleation and propagation. The nucleation, in case of written, you have to work with a small energy, that is the kinetically controlled. You have to fix only 2 phi and psi. Whereas in case of the starting with the alpha helix, you have to start with the 3 phi and psi. So you are not in a position to spend the money. So you will work with the 3. So you will start with the written helix. Then once the written helix is formed, you find no, he is poking his nose too much in my business. So then from the eclipsing, you go for the step and Gauss form. Okay. So this one is
thermodynamically favored. Alpha helix is thermodynamically favored.
Yeah, alpha helix is thermodynamically favorable, and that's why you are getting a long chain. So what you are finding this one, you see that in all the cases, there is no staggered, no eclipsing interaction between these purple balls. All are the side chains. They are far apart from each other. Suppose you have your friend, you have your family members, you have also your own identity. And so if someone is poking 100% time his or her nose in your business, you are not accepting that thing. So the eclipsing interaction is 100% time, one each another side. Okay, so it is energetically disfavored. Okay, initially it is okay. So what we are going is basically after that, between the secondary and tertiary structure, we find that there is another one that is called the super secondary structure, where there is a difference of the secondary structure oriented in a particular fashion. And one or two very good examples are Greek key motif and Rossmann fold, etc. So when we talk about the tertiary structure, we find that in case of the tertiary structure, we have basically the two. So secondary structure also, it is in between local form, but in case of the tertiary structure, it is a through space. So where your friend is staying in Khidirpur and you are staying in Bonhugli, in Calcutta University, you meet together. So this is a through space connection, you have a friend. So like this, they have, and there are different interactions, hydrogen bond, hydrophobic interaction, electrostatic interactions, salt-bridge interaction, disulfide interactions. So this is basically the protein data bank where you will get that thing. Devnapal from IRC, he may say you about the database. So here, this is the rcsb.org. If you put your search, suppose a myoglobin or oxygen carrying protein, whatever you put, you will get this thing. I just put this and I will get, you can get the second one will come. In this particular case, you find there are the download files or view files. So if you go to the download files, you can get the FASTA sequences, you can get the PDB sequences, whatever the thing. And for the defining of the secondary structures, you have to use this DSS. So in PDB, what you are getting only the protein coordinates are there. the C alpha, N, etc. coordinates are there. Whereas in case of the secondary structure, this is the DSSP. This is called the definition of the secondary structure of the protein or dictionary of secondary structure of protein. You can go to this site HTTPS and there is a web server. You just put any protein name or you just put any protein ID. That is the ID, you find that 2JHO. These are called the PTP ID. You can find 1J3F. These are the protein ID. So you just put there, you will find this. And you will find this is the secondary structure, basically on which they are saying H is alpha helix, beta sheets are there. So I am showing this thing. This is an example. So here you will find this is basically you will find PP1, PP2. SEC means surface. You are giving the secondary structure just one beta strand and phi and psi. So the difference between the PDB and the DSSP is that in case of PDB, you are getting only the coordinate, but not the secondary structure information, that is phi psi. Whereas in case of the DSSP, you will find this is someone. This is 1, 2, 3, 4, the serial number 681. 681 means the residue number. This is the chain A. The amino acid is S, means serine. Its structure is not there. But if you go to 683, this is lysine and it's an E. What is the E structure? You find E means strand, beta strand is there. And you will find that what is BP1, BP2, there is there. The surface accessible area is 9, means very small. So it is within the, but this is the NHO is there, and you will find there's a phi and psi. So if you see this thing, if you go in this way, I'm sorry, this is not a straight line, you will find this thing, and this is the XCA, YCA, and ZCA that is giving you the C alpha coordinates. So for a structural biologist, PDB is important because it will give you a structure, it will give you a help to understand this thing, but the DSSP will give you the residue-wise. So the question is, what determines fold? We are talking about too much. This was a novel work by Anfins and Sebi. In 1957, he did the experiment, and in 1973, he was awarded the Nobel Prize. And in his lecture, he mentioned in the last slide that the function of a particular system depends on its structure, and the structure depends on its sequence. So if you see the fingers like this, we can fold in a single or unidirectional. Why? Because the clampings in the finger in such a way, it is not allowing us to fold in an opposite direction. So the structure is important. And the structural information is basically placed within the sequence. That was given by Chow and Fashman, who said that out of the 20 amino acids, all the 20 amino acids have not the equal propensity. So some prefer the alpha helices, some prefer the beta. Now we have to go for the different way to understand what are the secondary and the tertiary structures of. So there are the experimental techniques. In case of the experimental techniques, we find that the experimental techniques are two types. One is the X-ray crystallography, and the second one is NMR. And there are computational techniques that there. In case of computational techniques, we have the knowledge-based approach, that is homology modeling, there is a threading, ITESR, and hierarchical methods are there. And why we have to do this? Because the prediction of the structure is important to understand the function. If you have a structure, if you know a sequence, it will be easier to predict the function. So people are working on that. So I am not going into that part of the bioinformatics, but I will show a little bit how the homology modeling and threading, because the molecular dynamics part will be taught, and he will tell you something. I will show you a little bit of experimental technique. There are experimental techniques that mainly initially people were down the X. This was the Kendrew and Perus in 1960 first provide the structure of the myoglobin. And before that 10 years, Linus Pauling in 1951-53 proved with a broomstick and thread what are the structures of the helix and the beta strand. That is nothing. Can you tell me why people are saying alpha helix? Why people are saying the beta strand? Because the helix was initiated or generated Linus Pauling first, and he took the first word from the Greek alphabet alpha. He put the word alpha here. Then he generated the model with the broomstick. Bangla bhashay jake bole jhatar kathi ar shuto diye model toiri korechilo, and that during that time genius works together here in India in IIT Madras, Chennai, IIT Madras. That time G.N. Ramachandran was working on the collagen helix. 1950, Linus Pauling, G.N. Ramachandran, all are giving their results. So the problem in X-ray crystallography is that there is no solute-solvent interaction. The molecule is free. But in NMR, which is invented much later in 1980 or after that, where you can get the solution structure and you can have an idea about the system. In case of NMR, I will little bit tell you NMR. NMR means the nuclear magnetic resonance. So the first term is nucleus. I have an YouTube in a channel. You can see what is the details of NMR. The basic part is there. So NMR nuclear means those where the I value is not equal to 0. So what is I value? I value means the nucleus is formed by the proton and neutron. If we consider the sum of the spin of the proton and neutron like electron, spin half value, we find that in the case of one H1 on the proton is that it is a spin half. There are three categories of the nucleus. One is I equal to 0 and the another one I is not equal. The I not equal to 0 can be divided into two parts. One is called the Hopf integral, where the 1H1, that is the 13C, these are important, and the other one where the spin value are integral, like the deuterium, oxygen, etc. So what we are getting? So these are the biologically important. Can you see the biologically important nucleus? The 1H1, proton, 13C, phosphorus, and nitrogen. People are doing with this. For the nucleic acid, people are working with phosphorus. For the protein, people are working with hydrogen, carbon, and nitrogen. So what is this? So NMR is a tech. It is a biophysical tool which gives you an idea about the structure or elucidation. It covers the study of protein-protein interaction. It covers the study, helps to identify interaction of a particular protein in a solvent, which is not possible in case of the X, because X-ray works with the freeze molecule. And this is basically in 1980s where the small proteins are working, basically the amino acid with the peptide. And in the present case, this is the magnetic field strength 500, 600. Now people are working with a gigahertz. I personally work with 900 megahertz machine in IIS Bangalore or TIFR. And now people easily can work with 100 kT protein. So they are giving rise to this structure. So the protein NMR, we have to have the 2D and 3D. This I am not showing this thing, but this is actually to show you that with age, with time, we are increasing, and so this is basically the length is increasing, the protein size is increasing, as well as we are increasing the structure. So how we are getting this? So this is the linear sequence. And if we consider this one, this is basically, this is the Broadway. So we have this is the NH, this is C alpha H, and basically all the tricks come here. So how we are doing this thing? So we find that this NH proton, this is the one D structure, the NH proton comes in the region of 8 to 9. This is basically taken from Utrecht, and I am fortunate enough to see the Utrecht in somewhere who was in 2002 got the Nobel Prize, and basically of this work, and this is basically taken from the Utrecht's book. So you find that this is the basically the signature. This is the NH backbone. It comes around 8 to 9. This is the NH side chain, that is lysine, etc. It comes around 7. This is the ring, that means the aromatic structures that they are, which I am giving it here. This is the ring structure that will show. Now, in the case of alpha, C alpha, these are basically coming here around 4.5, 4.7, like this. So 4.3, 4 to 4.5 within that region, even 4.6. But the serine and threonine, this beta structure, beta protons are also coming here. And other beta, gamma, delta, these are coming in this. And if you see that, this is called the chemical shift value, that is depending on the frequency on which it resonates. So when you find that the higher field, you consider in this thing. Upfield means if you want to see your friend, consider in your young age, when you are going to your school and you are looking, you are waiting for your girlfriend, who is, it will be easier if it comes in veranda and you just put a single ring in the cycle. Here you can see her easily. So you have to give low. So this is called downfield. That means they are not covered. But if your girlfriend is within the room, she has to come out. So you have to give more number of rings. So that means your field strength you have to increase. That means if a proton is shielded by electrons, that is, if the OH is there, the oxygen withdraws more number of protein, nitrogen withdraws more number of electrons from the protons. So they are bared, they are open, they are exposed, and you can see them. But in this particular case, you have, if you are CH3, where more electrons are covered, so you have to give more amount of effort field strength, and that is basically the beauty of the NMR, for which you can distinguish. And this is called the chemical shift value. For higher field strength, your chemical, that means where you are having, you have to give more impact, you have to give more input of the field strength, it will resonate in the low field. That is the low delta value, chemical shift value is low. Whereas in case where you have to give a small field strength, that is called the down field, it is coming at the higher. So this is basically the best on the chemical shift. And now, depending on the chemical shift value from the 1D structure, there are few methods that there. One is called the chemical shift index. And this chemical shift index, there's a random coil, Utrecht measured. This is the glycine, some particular residue and glycine, because glycine is helix breaker. It is a random coil. The X residue will never be helix. So he found whatever the things, he got this thing, and he called it the random coil. And with respect to your random coil, when you are taking, if this is the down field, then you find that it is the beta stand. That means positive values are. But if you find in the upfield, that means it's a negative value with respect to random coil, you call it a secondary structure helix. So chemical shift is an important signature through which you can measure the secondary structure. The second one is the temperature coefficient. That will tell you how many protons, how many are solvent exposed. This solvent exposed number of proteins, if in case of alpha helix is a 3, in case of written helix these are 2, in case of random all are exposed. So the value is delta delta T, delta delta by delta T. If it is less than 5 ppb per K, you can consider it an helix. And now the number will give you this, and more than 7 you will find that is random coil. And the second one, and the third one is the coupling constant. Suppose I am close to you, I can talk to you. And this coupling constant, what I told you, this one, this NH and C alpha H, they can talk to each other depending on their geometry. And that is basically coming from the TCO, which we have seen in the DSSP structure. We find that thing, this is, if these values are basically 4 to 5 hertz, we call it helix. If it is more than 8 hertz, we call it beta strand. In case of this is random point. So what we understand basically from this, depending on the particular phi and psi value, we are having basically particular dating shape. And once the particular shape is generated by a particular secondary structure, then the residual, whether the hydrogen bond is formed or not, we can see. So whatever we are getting basically the secondary structure, which is the key of the formation of the tertiary and the quaternary structure, they are having coming from this of the phi psi values. Why phi psi values? There are 3 dihedral angle omega, phi, and psi. Omega is used either 0 or 180 degree, that is used for cis and trans for the primary structure. And you can see the 2-dimensional structure of the phi and psi giving the 3-dimensional representation. So this is basically. But we have 1D NMR structure. Why 1D? We are plotting frequency versus intensity. But the 1D is not visible to get the protein structure. Why? If you see this thing, these are very much jumbled. These are, from this it is very difficult to identify which one is what. So we come to the next one, which is called the extreme overlap, unable for the structural characterization. So we have to go for the 2-dimensional structure. And this 2-dimensional structure, we have to depend on 2 ones. One is, suppose there is A, B, C, D, E, 5 persons are there. One we call the scalar coupling or through bond coupling. A and B are connected by hand and to hand. So A or B, they are basically through bond coupled. But A and E, they are not through bond coupled, but they are coupled via B, C, and D. But somehow they may be close to each other, and this is called through space. Through bond couple, for this we are doing the NMR, that is called the COSY, correlation spectroscopy. This is called 2-dimensional correlation spectroscopy, and we find that there are cross peaks. We find that here these are the 2 peaks are there, and you can see these are the 2 cross peaks. So the diagonal peaks, these are the straight lines. So this is the diagonal peak. You find that this is the diagonal peak, and between the diagonal peak, 2 peaks are there. And if they can talk to each other, so you will find basically a cross. So this is like a square. So here you can find this. So what is basically you find in this particular case, this is the diagonal peak, and here these are the 2 cross peaks. That means they can talk to each other, and that means they are through bond correlated. So if we go NH and CH alpha, so if we go like this, N, this is C, and this is C, so this H is coupled with this H. This is coupled with this H, and this is coupled with this H. That means this is coupled with this H, and this is coupled with. But these 2, if you see this thing, sorry, if you see this thing, these 2 are not coupled to each other, but these are coupled via this. These are coupled via this. Okay, so this is called the 2-dimensional spectrum. Now, in case of the protein, so what we are getting, if you can remember this thing, this is the NH region, This is basically, this is the NH region. This is the diagonal peak of the alpha region, and this is the beta. So what we are getting, this is the cross peak we are getting between the NH. This is the cross peak we are getting between the NH and C alpha H, and this is the cross peak we are getting between the NH and C beta. So this is the region we are having. So we have to look into this. And now in this case of this folded structure, so once the protein is folded, you find that these two are very close to each other. These two are very close to each other, so they can talk through space. But if they are unfolded, they are so far apart, so you are not getting it. So how do you understand this thing? So the next one, which we will get the spectrum, it is called the NOE or the gnosis. That is, NOE means the nuclear Overhauser effect. So nuclear Overhauser effect means it is related to distance, and this relation is R to the power 6. If this is going here in this particular, if they are related by less than 4 angstrom, if this is related by less than 4 angstrom, we have this NOE. If 2 angstrom, 2 point something, 2.8 or something, we get a strong NOE. If it is 3.5, we got a medium. 3 to 3.5 is medium. If it is around 4, we got a weak interaction. So what is, depending on the strength, depending on the volume of this thing, actually we are getting a peak like this. It is like this, that this is the 2-dimensional chart. So depending on the contour map, we are measuring what is the volume. So this is basically a structural skeleton where we found the NH and this NH, they can couple for the structure. The C alpha H, NH can couple. This is the residue with I plus 1. So when we are talking about this is the I plus 1, we are getting this thing whether we find this or not. If the structure is helices, we are having this thing. If the structure is not helices, we are having this. Moreover, there is one more important thing, is the alpha N NOE walk. I do not know whether you have done this thing. Suppose you see this is residue number 2, okay, and this is residue number 3. So what we find that this is the residue number 2, this one. Then it starts, this is the original peak of residue number 2, and then this is the alpha region, and this is the N region. We are getting a cross peak. So it is, you find there is no cross peak between this. This is no cross peak between this. So what we understand, that means they are related like this, I to I plus 1 related. So we find that if this is 2, obviously this is 3 and this is 3. Then you find 3 and 4, this is the peak between 3 and 4. And 4 and 5, you find that this is the peak. This is the peak between 4 and 5. So it helps you to walk and to identify who are with who. A is connected with B, B is connected with C, C is connected with D, D is connected with E. So this is for the sequential walk, and that will give you the amino acid sequence. So this is called the sequential walk, what I have written here, I to I plus 1, NN I to I plus 1, alpha N I to I plus 1, beta N I to I plus 1. But now you have to go for the long range, that is giving you the structure. If you have with this NNII plus 1 and alpha NII plus 3, then you can call it is a helical structure. So connectivity of this, say this thing. So connect C alpha H residue versus this one I NH of I plus 1. This is the beta H, so it is for the sequential walk. And then you go for this one, you find that this is, see this thing for helix, alpha helix and 310 helix. You see NNII plus 1, both are positive. You see both are positive, I plus 3. I plus 3. Both are positive. You find I plus 3 are found. I plus 3 are found. But in case, how we can distinguish? In case of the 310 helix, I to I plus 2 is present. But in case of alpha helix, I to I plus 2 are present. So in case of helix, whether you first to identify, so it is basically I to I plus 1 and I to I plus 3, and then you have to go for this. So this is one example I am showing because the time is not more. So this is basically from the 1D. This is what I told you about my proteins paper. This is a structure, and from that we generate this thing. From the NMR structure, NMR data, we can have this structure. This is the basically we are getting the helical structure depending on some software called Dyna, and now people are using Sina. Now, the last part, 2-3 slides, how to predict function. So once you have two proteins, you know the sequences. So what you have to do? You have to map the sequence. Why? Because you have to consider the function depends on structure and structure depends on sequence. If you have two human hands, you can easily match together. So you find that they can walk in a similar way. But if you have one cow and one human leg, you can easily find that they are different. So the sequence information is very important. And then sequence to structure and the structure to structure fold is there. There is called the superpose. And this is the superpose. This is you go. There are two PDB structure. You give them. The server is there. They will give you all this. So there is a term called RMSD, root mean square deviation. If the root mean square deviation is very low, that means you are in a good. This is the last part. I am showing that there is a homologous protein. So homologous means they have the same ancestor, like the 5 fingers of the hand. It is a quite interesting, and recently I have published this paper. We find that out of the 5 homologous protein, PPCA is interacting with DXCA, but its other homologs BCDE doesn't interact. So when we match the pairwise alignment, we find that interaction region of the PPCA and versus PPCB, this is called the structural, this is called the sequential, pairwise sequential alignment, and we find they are different. And when we superpose this structure, we find that this one, the red one, this is basically, you find that this red one, and the black one is the BCDE. So we superpose, we find that everywhere only this A is basically helical, and other cases this is not helical. That means the secondary structure is an important role, playing an important role in the function. So what we understand, what I am going to tell you is that is the sequence gives you a structure, and the structure gives you the function. So from this, from the very nutshell, we can see this one, the PPCA, which is interacting with the DXA, is sequentially little bit different from its other homolog. At least you find only one sequence is different here, G2. Here you see G2S is only different. Others are same, and K2V is different. So it is not interacting.
Dear all, please stay connected. Sir has disconnected due to internet issue. So sir is joining. Please stay connected. Soham sir will be joining very shortly. Sir has some internet issue. He has a rejoining. Okay.
Okay, bhaiya.
Yeah. Dear all, please stay connected. Yes, sir. Your PPT is visible, sir. Sir, please unmute your mic, sir.
Can you hear now?
Yes, sir.
Hello?
Yes, sir. Yes. Please, sir. Continue, sir.
Okay. So if you see this thing, the sequence alignment, which is done by the cluster W and cluster omega, who are working with this, the A and B, you see that I is important. It is changed to M, K is changed to B, but others are M, G is changed to S. So there is a mutation. What yesterday was telling my professor there. So the mutation takes place. Similarly, I to I is there, K is changed to G, or K similar to R, but because this is the amino acids and all other almost similar, only these two changes, A to G, makes the PPAC different from PPCA. So here,
Sir,
if you see
voice
this
is,
thing,
sir, voice is very low, sir. Yes, sir.
hello,
Yes, sir.
can you hear? So if you just superpose these two PDB structures, if you compare this comparative structure of PPCA, which is in the red, and PPC B, C, and D, and E, which is in black in all the cases, you find that in PPCA, this region, which is the highlighted region, these are helices. Whereas in other case of B, C, D, and E, you find that they are almost random coil. So from that, it is very important to note that the secondary structure plays a very crucial role for the particular function. So the binding, the protein-protein binding depends on whether you can adopt a particular structure or not. Just like if you have a 5 finger, you can easily catch the thing. If you have not this thing like this, you cannot catch the thing. So you can, if you want to hold the thing, if you have to catch a ball, you have to have the finger instead like this, a ball type. And that is a nest where the eggs are there. It has to be like a bowl shape. So similarly, the structure is an important role. So the last part of this is, at a glance, what we can see is sequence is a very important thing. We have to start just the opposite reverse way of the Anfinsen Seavey, the pioneer legendary work and his last word of the Nobel laureate Nobel lecture in 1973. The function of a protein depends on the structure, and the structure depends on sequence. We go in a different way, and it is Nobel Kellenberg who said the protein folding from the first principle. The sequence gives the structure, and the structure gives the protein. So that is the last word we have to say. And this is, thank you for your kind patience. I'm extremely sorry, some interruption. So any question, any queries, any suggestion? I hope I keep the time.
Hello, sir.
Yes.
Myself Milan. Actually, in your presentation, you show that mRNA run in backward direction during
Yes, that is
protein
called reverse.
protein synthesis.
That is
Yes.
called reverse transcription.
Okay. Now, in your protein machinery during protein synthesis, mRNA run back. In between ribosomal subunit.
Protein, I do not understand your question. Protein synthesis.
During protein synthesis, you show that mRNA stand run back inside the ribosomal subunit.
Okay. That is the tell me.
But it is always true that mRNA only run back. That a ribosomal subunit never move over the mRNA unit.
I do not get your point. You are asking about that movie slide.
Oh, yes.
You are going to movie slide. Okay.
Yes.
Where you are asking? I don't get your question.
Protein machinery during protein synthesis, mRNA stand only run. Where the mRNA stand run in backward direction.
In prokaryotic cells, translation is initiated by
you ask me to
formation
stop.
of an
I will stop.
initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit.
So here you find that there are the mRNA is there, okay, with the codons. And the tRNA is coming, but when the tRNA is coming, there are the 3 RNA units are there, okay? This is the mRNA part, this is the tRNA part, and this is the ribosomal subunits are there.
Yes.
Okay? So when this is coming, this is the first, that is called the start codon, AUG. That is taking the phosphorylated unit. Okay? But how this will work? This is the 30S and the 50S that will together come together to make a complex, and that once that will start this two.
In prokaryotic cells, translation is initiated by formation of an initiation.
What is said? Translation is initiated. Learn this word. The translation is initiated by the formation of, now see,
complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA.
So if we stop this thing, once the 30S ribosomal unit is coming on the AUG part of the mRNA, then the AUG is activated. Okay. Then it asks, basically, suppose if you are in the age of 4 years, you are not looking for your some other opposite part. But once you are at the time of your adolescence, what you are doing now, you are thinking in a different way. So that means you need add some. This is something. If I use the word epigenomics, it is wrong term. But you need an environment. So what environment is this? This 30S ribosome unit which all comes from the mRNA that activates AUG, which calls the UAA anticodon tRNA with the formilated methionine. Am I understandable?
Yes, sir. But my question was that mRNA stand only move, or
Yes.
ribosomal unit also move over the mRNA stand?
No. That part is fixed. I see that. You see this.
Okay.
You see this. You see this.
Eukaryotic cell.
Okay.
I understand that.
Evolution is initiated
Okay, sir.
by formation
I got this
of
part.
an initiation complex.
Actually,
consisting of
mRNA
30S
moves on the
ribosomal
P side to
subunit,
E side. See this. See this. See
formylmethionine
this.
tRNA, and
Yes,
messenger
yes.
RNA. The 50S ribosomal subunit then
This
joins
is the
the
first.
complex.
And
Proteins
once this
called
50S
initiation
is joined,
factors are
then the
also
three
involved,
sides
but
are
are not shown.
activated.
The
P side,
70S
A side, and E
ribosome
side.
has two sides to which
Yes, sir.
transfer RNA carrying amino
Now
acids
mRNA
belong.
moves.
One is called the peptide or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid and moves it to the A site.
So you now activate the site.
The
Okay.
formylmethionine
Now you
carried by the transfer RNA in the P site
see
is
this.
then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer
Do you understand
RNA
now?
carried the formal
Yes, sir. Yes, sir.
is
I got it.
released.
Now, wait, see this.
A transfer RNA.
The whole
carrying the
complex
next
is
amino
moved.
acid
Once the
moves
P
into
site
the
is there, A
where
site
the
is activated.
anticodon on the transfer
See
RNA
this thing.
matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As
Okay.
the shift occurs, the two
Yes, sir.
amino acids
Sir,
on the
one more
transfer
question
RNA
from
in the
Ramachandran plot, where
Pardon?
you mentioned the regarding Ramachandran plot,
Yes.
where you mentioned the psi angle and phi angle, that are the torsional angle. So this angle is possible for the isolated amino acid, or it is only possible for the complex peptide in case of peptide?
No. This is for the actually what G.N. Ramachandran has done. G.N. Ramachandran has studied the proteins. Okay. And
Yes.
he has calculated with different distances how the amino acids are there. And then he plotted the amino acids. They are phi side. Okay.
Okay.
He found in this particular case, what he found is the mostly alanine are present here. Okay.
Yes, sir.
And what he found
Okay.
that the phi and psi, that time he didn't have an idea phi and psi. He was started his calculation with the distance. That is the Van der Waals contact. If I am sitting on your lap, it is difficult for you to carry me. But if I can see at your side, you can talk to me, isn't it?
Yes,
So
yes,
that
yes,
is
yes.
called the allowed and disallowed distance. Okay. So Ramachandran
Yes, yes.
and Shashishekaran in 1966, they have started with calculating how they are allowed to what distance. And in
Yes.
case of collagen, based on the distance parameter, they are framing this. And after that, they form the calculation of the eclipse and staggered interaction. Based on that, the energy versus the plot. And then they use the term phi and psi. Okay. So then
Yes,
they
sir.
whole represented. আরে এটা কি হয়েছে? এটা পুরো ব্যাপারটাই তো একটা phi, psi আর omega, phi, psi আর omega. If you go to this thing, if you see this thing, there is nothing. See this thing. MC alpha psi. Okay, phi. This is psi. This is omega. This is phi, psi, omega. Phi, psi, omega. So the whole linear sequence, you can have the three dihedral angles, phi, psi, and omega. But
Yes, sir.
এইবার যেটা হচ্ছে, if you consider your wall of a house, your length of one wall is 6 feet, suppose. 6 feet is long, so that means there is no change. But after the 6 feet, you find that there is a change and angle become 90 degrees. So that means this dihedral angle comes there. So the first 6 feet, this is a linear sequence.
But after the 6th bit, there is a kink, there is a turn, there is a torsion, and this is you are using the secondary structure of that. Then you are making it as a square form. Understood?
Yes, sir. Yes, sir. Okay, okay.
So you can go like this. You can go like this.
Okay, sir. Okay, thank you, sir.
I think you can go like this. So this is the linear structure, 6 bit. There is nothing, only omega is there. But once you find that there is a wall, you cannot move. So what you have to do? Before that, you have to now a turn, isn't it? Municipality bole diyeche 4 foot রাস্তার আগে ছেড়ে দিতে হবে. Okay? So you have to bend that. And then that will create a plane, and that is the torsion angle. And this then comes about the phi and phi. Okay? Otherwise, up to this, there is nothing, only omega. Okay?
Okay, sir. Okay.
Any other question? So, is there any comment, any suggestion? At least one or two before closing? Can I make you at least little realize the whole part of the protein structures?
Yes, sir. It was very insightful and wonderful lecture.
So may I know your name, please?
Sir, I am Shravani Kormakar from Techno India University.
Oh, you are talking about that you work with the protein structure, function, etc.
Yes.
Okay. So where do you work? On which platform?
I'm in Techno India University. I'm teaching in.
No, I am asking about your research.
Oh, research. Sir, I have one pair project in which actually I'm working on alpha B crystalline. It's a chaperone protein, and yes, so the project is about actually some mutations which are naturally occurring in myopathies, cardiomyopathies. So in the project, we are aiming to look at the structural differences and how it relates with the chaperone function.
So you know all these things, what I have told you today.
Yeah, NMR part I didn't know, sir. And yeah. So
little bit of this structural Ramachandran plot and this energy part, I think you know this.
Some parts are known to me, yes.
Yeah. So any other? Madam Ruhi is here. Yesterday she was talking. Or Iqbal, who is an information scientist, or some others. Any suggestion?
Sir, I'm very much delightful after learning all these things from you, sir. And it is really a good opportunity. This is the first time I'm listening you. I'm impressed. And actually, I would also like to work on this bioinformatics and all this. And these stuffs are very interesting. And learning from this basic point of view is very nice. And I have enjoyed this whole 2 hours, sir.
Truly, or
you presented the whole thing, the whole big thing in a very peaceful way, so that we can easily understand. And there is suggestion is there is no suggestion as per sir. You are enough. I'm very happy. That I can say only. And in future, maybe I can utilize these things to take some suggestions from you to do some activity related to this, sir.
but give me some criticism. I want criticism. So, sir, this is not good. Yeah, you have to do these things more positive way. I want criticism.
No, sir. Not at all, sir. Not at all, sir. I'm from IT department, but learning this, I don't think that it would be much easier to learn understandable. As far as I'm concerned, I'm little bit lesser knowledgeable in the field of biology. So still, I found it's interesting. So I think this is enough, sir, that you have taught us well. There is nothing criticizing you, sir.
Okay. So, Madam Ruhi is there. You are from Kashmir. I'm quite impressed that you have joined. Hello. Ruhi Madam is here or not? Same?
Sir, ma'am is disconnected, I think. Due to internet issue, ma'am is disconnected.
Okay. So, Mr. Tapan Kumar Pal. Are you here, Mr. Tapan Kumar Pal?
Yes, sir. Tapan sir is there. Sir, please unmute your mic.
I'm hearing.
Hello, sir. Hello, Mr. Tapan Kumar Pal.
Yes, I am hearing.
Okay?
Yeah, it is okay. It is okay. It is okay. I am seeing the Ramachandran plot. It's okay.
Okay. And Madam Karobi Ganguly is there?
Yes, sir. I'm here. The lecture was very informative and resourceful. But who are a person like me who are not directly working in this field, we need a few more working on this to assimilate the whole thing. It was so resourceful, so informative. I think many other my batchmates, my colleagues who are attending this STTP, they are partially or having some knowledge or work experience in this domain. But I am totally new in this field. Your lecture was very, very informative. But I have to go through this at least 2-3 more times, so that to assimilate that thing.
I think this will be in the YouTube, Prem.
Yes, sir. Yes, sir. Yes,
This
sir. We
will be
will
in
provide the
YouTube.
video link. We will provide the
This
video
will-
links.
yeah. So you can see. But actually, my point, if there is someone who do not know or who doesn't know anything, at least he can have a 10% idea about the protein structure. At least he or she understands what is the dihedral angle, what is the difference between the sequence, and how the sequence can play in the role of structure. Because if you can remember only this thing, if you have an alanine, if you have a leucine, you can frame a alpha helix. If you want to generate a beta strand, if you have to prefer for the isoleucine or phenylalanine. If you want to break a helix, you have to put a proline. At least if you can remember, if you can have this idea, I consider myself as a protein tailor. Just like we have to design carpenter, we are doing this thing. So this carpentry gives you some basic idea, and if you have this basic idea, if you have this basic knowledge, I'm telling you very honestly, no one taught me these things. I learned, understand this thing during my research and throughout my life. So I wanted to make the whole thing in a comprehensive way so that you can understand what is protein, because the 11 players who are playing in the field, they play the key role. If the goalkeeper is in the striker position, there is a mutation. And the striker is in the goalkeeper position, the fate of the football team, what is going there, the similar to that would happen in case of the protein. I understand this in this way. If Harbhajan Singh comes in place of Sachin Tendulkar, the thing of the Indian team, what will happen during the batting, and if the wicketkeeper goes for bowling, the opening bowling, the thing, what will happen, that is called the protein mutations. I understand the whole thing in this way. Okay, so this is my part to let you know. And if no one or someone who has no idea about this thing, if you can little bit have an idea about the protein structure, I feel myself thankful. This is a duty of a teacher, is to tell you some stories, some thing about this. And if others enjoy this thing, I am happy for them. So thank you very much.
Thank you very much, sir, for your self-explanatory lecture. It was awesome, and not exactly 10%. I think the whole batch have received information up to 40, 45%, 50%, and the next remaining part we have to work out.
So next, from 5 onwards, there is a big person from computational data science of IISc Bangalore. He will come. So please be here by 4:50. Okay. So then you will understand what is data science and how it will go and how you have to look at these things. Then the big data, from the data to big data. So he will take you in this way. Actually, I can tell you the data best, but I do not tell you the database. Why? Because it will be easier for you to understand how the data to big data from the same person. Okay. So thank you. Have a good day. We will meet again at 4:50 in the morning.
Mute your mic. Sir, please